Good evening, students. So in the last class, I was explaining genetic code, and in the last class, we were talking about a brief introduction about the genetic code, and even we discussed about the types of amino acids, like essential amino acids and non-essential amino acids. And dear students, while explaining the names of amino acids in the last class, there was some error, writing error. Kindly, I apologize for the mistakes that I made while writing name of the amino acids. Okay. So today we will be discussing about the properties of properties of a genetic code. Right. In the last class, we discussed that there are 64 genetic codes are available. Is it? And even last class I said that how to write. Okay. So each genetic code will be having a 3 nitrogen bases. First nitrogen base from this side, second nitrogen base from here at the top, and third nitrogen base is from the right side. This is how we can write all these 64 genetic code. Okay. And even I said that there is a trick, there is a mnemonic to remember this genetic code. So today we will be discussing what are the tricks, what are the mnemonics to remember all the genetic code as well as all the amino acids. Okay. And if you see this table, there are 4 columns, isn't it? Column 1, 2, 3, and 4. There are 4 columns. That is what I have written here. Column 1, 2, 3, and 4. So we will try to remember all these amino acid name as well as a genetic code column basis. Column 1, 2, 3, 4 like that. We try to remember. First, you take a column 1. First one, column 1. In a column 1, how many amino acids comes? You just see. Phenylalanine, leucine, isoleucine, methionine, valine. So to remember this, there is a trick. Prolearning in, or you can take instead of in, you can take is, then it will be very easy. Is molecular valency. Okay, so this is a method or trick to remember all the amino acids of column 1. So what is the meaning of this? After writing this statement, here you write 2, 6, 3, 1, 4. So we should remember numbers also. So what is the meaning of this statement and what is the meaning of that number I have written there? See here, I have written a P. Here P means phenylalanine. Okay, and why I have written here 2? Here I have written 2, right? It means first 2 codon. In first column, first 2 codons UUC, they code for phenylalanine. That's why here I have written 2. Okay, this is correct. Now next one I have written a 6, learning. Here learning means leucine. So here L stands for leucine. And why I have written here 6? Because after 2 phenylalanine, next 6 codons. See, 1, 2, 3, 4, 5, 6. So these 6 codons are coding for leucine. So that's why I have written here 6 learning. Now next here I have written that 3 is. It means 3 is means isoleucine. Okay, so it means isoleucine. Why this number 3? See here, 1, 2, 3. 3 isoleucine. Next, here I have written one molecular. Here molecular M means methionine. It is methionine. Right? So how many codons are there for methionine? See here, only one code for methionine, that is AUG. Okay. And finally, 4 valency I have written here. So what is the meaning of 4 valency? Here 4 valency means valid. See, 1, 2, 3, 4. So this is how you can remember all the codons and all the amino acid names in a column 1. Still, there are 3 columns. There are 3 columns are there. First, you be thorough in this first column. Okay. To prove 6 learning, 3 is 1 molecular or plants. Now let us see second one. Second column. Let us see column 2. This one. And to remember second column, again there is a trick. Cereals profit. Cereals profit through Alia. Okay. So just imagine there is a cereal producer. He want to get a profit from his cereal. That's why he planned to introduce Alia. But by remembering this statement, you can easily recall all the amino acids. How? Before that, write every day 4. So here, first you take a cereals. Cereal means cereal, serine. Serine. And how many codons are there for a serine? 1, 2, 3, 4. That's why here I have written 4. So there are 4 codons for serine. Next you take, again here I have written 4. And I have written profit. So here profit stands for another amino acid. Name of that amino acid is proline. Proline is another amino acid. And you can see here how many codons are there for a proline. 1, 2, 3, 4. That's why here I have written 4. Now let us see true. Right, again 4. Here true means 309. See here 309, 1, 2, 3, and 4. So how many codons are there for a 309 amino acid? 4. And finally is Alia. Again, same 4. And here alia stands for alanine. Okay, see here, alanine. How many codons are there for alanine? 1, 2, 3, 4. So that's what here I have written 4. So how to remember all the genetic code and the amino acids of second column? Very simple. Cereals, profit, true. Cereals means serine, profit means proline, true means threonine, and aria means alanine. So this is about second column. Now let us see third column. Next one is column 3. Okay. And again there is a method or trick to remember that. There is a boy, his name is a Taylor. There is a boy, his name is a Taylor. Taylor stopped. Taylor stopped his giant. His giant ASN bike. Just imagine there is a boy, his name is a Taylor, and he has a very big ASN model bike. Where he stopped? He stopped this at ladies' store. Why? To buy aspirin tablet, and glue, or gum, you can say gum or glue. Okay, this is a trick we can use to remember the amino acids of third column. Okay, Taylor stopped his giant ASN model bike at a ladies' store to buy aspirin tablet. Actually, it is and you have to write for every word. You have to write 2, 2, 2. This one is not necessary. 1, 2. Now let us see what is the actual meaning of this statement. First, here I have written Taylor. Right? In fact, I have written to Taylor. It means Taylor stands for tyrosine. See here, tyrosine. And how many codons are there for tyrosine amino acid? 1 and 2. So that's why here I have written 2. So Taylor means tyrosine and there are 2 codons. Now next, stopped. This one is a very important. Stopped. See here, soon after tyrosine, here in a genetic code table they have given a stopped. These 2 codons they are called as the stop codon. Okay, what they are called as? They are called as stop codons. Okay, why they are called as a stop codon? You will come to know later when we will be discussing about properties of genetic. Till then, you just remember that these 2 UAA, UAG, they are called as the stop codon. How many stop codons are there? There are 2 stop codons. That's why here I have written 2 stop. Right? So as it is, stop means stop codon. Okay. Now after a stop, his. See here, his. Here, his stands for histidine. Histidine. Okay. And last time in the last class I had written this as a histidine. Sorry for that. It has to be histidine. Okay. So see here, histidine coded by 2 codons. That's why here also 2. So after histidine, and next it is a giant. Giant. So here giant stands for glutamine. It stands for glutamine, right? So here I'm writing 2. Why? See, GLN means glutamine. So there are 2 codons for glutamine. Next, ASN bike. See here, ASN. What is the meaning of ASN? You know that? Asparagine. It is asparagine. Okay. ASN stands for asparagine. And how many codons are there for asparagine? See here, ASN 1 and 2. There are 2 codons. That's why again here we have to write 2. Okay, asparagine is completed. Now ladies. So here, ladies store. Ladies store stands for lysine. Lysine, amino acid. And how many codons are there for lysine? Again, 2 only. One specialty of this column is, what is the specialty of this column? Every amino acid is coded by 2 codons. For tyrosine, 2 codons. For histidine, 2 codons. For glutamine, 2 codons. For asparagine, 2 codons. Lysine, 2 codons. Aspartic acid and glutamic acid. For every amino acid, there are 2 codons. That's why here, everywhere I have written 2 to show that there are 2 codons. Now, so here again, lysine also coded by 2 codons. Then this 2 by has no meaning. Now aspirin here I have written no. Aspirin tablet. Here aspirin means aspartic acid. Aspartic acid. Right? Aspartic acid. See here, ASP means aspartic acid. How many codons are there for aspartic acid? Again, it is 2. Finally, glue. What is the meaning of glue? It is glutamic acid. GLU stands for glutamic acid. Okay, GLU. Don't be confused. GLU, glutamic acid, and GLN, glutamine. Okay. So this is all about the amino acids are in the genetic code of 30. What is that? Yes, the last column of the genetic code table. And to remember this, we have again a trick. Cis, stopped, they tip. Cis stopped, they tip. Why? For argument, very serious argument, on glycine. Glycerol, you can remember this as a glycerol as well. So just imagine there are 2 sisters, okay, and sister, we also call as a cis, right? So here for our convenient of understanding, for our convenient cis, I have written CYS, okay. So there are 2 sisters, they are on trip, and you know that whenever 2 sisters be together, they never talk, they only fight, and they will argue. So there were 2 sisters, and they were on a trip, and they started arguing, and that argument turned into a serious argument, and they stopped their trip. And by the way, they were arguing, they were fighting on a concept. What concept they need? They had a discussion on glycerol. right? One sister, she was saying that glycerin, use of glycerol is very good, and another sister, she was saying that no, glycerol is very bad. Their discussion turned into argument, and because of serious argument, they cancelled their trip. So this statement is helpful to remember the support. Okay, how? See here, cis. Cis means cysteine. Okay, here cysteine. You can see here the last column, the first two codons, they are coding for cysteine amino acid. Okay, and how many codons are there for cysteine? 2, right? That's why here I'm writing 2. 2, 1, 1, 4. 1 second. 1, 4, 2, 2, 4. So this is the statement we can use to remember. See here, cysteine. Their first amino acid is a cysteine. And how many codons are there for cysteine? 2. That's why I have written here 2. And next, what is it? 1. Stop. Okay. Why here I have written a stop? You can see here. Soon after 2 cysteine amino acid, another one is stop codon. And students, so we already discussed 2 stop codons here. 1, 2. Now in addition to 1 and 2 stop codon, there is one more stop codon. Don't get worried. You will come to know what is a function and what is the meaning of calling them as a stop codon later. But at present, we should know there are 3 stop codons. Okay, here 1 stop means stop codon. So after a stop, trip, right? There I have written a trip. So here trip means tryptophan. It is tryptophan amino acid, as you can see here. Tryptophan. In an abbreviated form, it is called as TRP. Then, so here argument stands for an amino acid. The name of the amino acid is arginine. Okay. So what is the name of that amino acid? It is arginine. See. By the way, how many codons are there for arginine? 1, 2, 3, 4. That's why 4 arginine. Right? And after argument, again here I have written serious argument. Here serious. I have written 4 serious. Here serious means serine. Here serine. How many codons are there? 2. See here. There are 2 codons for serine. Okay. And again, after serious argument, here 2 argument again. You already know that what is the meaning of argument? It is arginine. Okay. And finally, 4 glycerol. 4 glycerol. So here glycerol stands for glycine. See here, glycine. How many codons are there for glycine? There are 4 codons for glycine. That's why I have written 4. So this is the statement you can use to remember all the genetic codon and amino acid given in the column 4. 2 sisters stopped their drink for having an argument, very serious argument on glycerol. Okay, so this is how you can remember all the amino acids and all the codons given in this table. Okay, so why we are this much bothered about this genetic code table? In a NEET examination and particularly in an AIMS examination, they may ask this genetic code questions in any way, like that. They once in exam, they ask that which is the only one amino acid that has only one codon. One codon. The answer is tryptophan. There is one more amino acid methionine. This is methionine amino acid and tryptophan amino acid. These are the only two amino acids which are having only one genetic code. If you want, you can search. Except here for methionine and there for tryptophan, you will find no way this is methionine. This is a way of asking question in examination. Okay. And once they asked a massive following type of question. One side they had given the codons, another side they had given this name of the amino acid and they asked for matching this. Okay. So that is all about genetic code table. Now let us discuss properties of genetic code. Right? So now we need to discuss what? We have to discuss the properties of genetic code. And students, in theory examination, this is the possible 3 or 5 marks question. Okay, many times they are even asked for 1 mark also. Let us see what is the meaning of properties of genetic code and how many properties are there in a genetic code. The first property is genetic code is triplet. Genetic code is triplet. Okay, it is the first property of genetic code. The meaning of this statement you already know in a previous class that each genetic code is made up of any 3 nitrogen bases. Totally there are 4 nitrogen bases. Out of 4 nitrogen bases, combination of any 3 nitrogen bases, is called as codon. And how many nitrogen bases are there in a codon? In each codon there are 3 nitrogen bases. That's why it is called as the triplet. And who was the scientist gave this triplet codon? It was George Gamow. We know that. George Gamow. He was the first person to give an idea of triplet genetic codon by applying the permutation combination method. He said that there are 4 nitrogen bases. Now, out of these 4 nitrogen bases, a combination of any 3 nitrogen bases will called as one codon. Okay, and in a given last class I said that totally how many codons are there? 64 codons. Okay, so if you want, you can count here in this table. You can count. You get 64 codons. And again in a 64 codons, 61 out of 64, 61 are called as the sensible codon, and 3 are called as stop codon. Of course, there are 64 codons, but not all codons code for amino acid. Okay, there are 3 codons which do not code for any amino acid. Such codons which do not code for any amino acid, what they are called as? They are called as the stop codons. How many stop codons are there? 1, 2, 3. Okay, and what is the meaning of stop codon? They do not code for any amino acid, unlike other codons. And why they are called as a stop codon? These are responsible for stopping, ending translation process. What is the meaning of a translation? Protein synthesis. These 3 are responsible for stopping, ending protein synthesis. Since they stop, that's why they are called as a stop codon. And students, it is the one of most common 1 mark question. give an example for stop codons. What are the three stop codons? One, here I'm writing UAG, right? See, UAG, UGA, UGA, and UAA. These three are stop codons. They have got another name. They have another name. They are also called as nonsense codon. They are called as nonsense codon. Why they are called as a nonsense codon? Because they do not contain any information about any amino acid. Whereas other codons, they have information. For example, let me tell you, for example, CUU. Whether it is a stop codon? No. Why? It has information about leucine amino acid. To take this one, ACU, it is a codon. It is having information about the threonine. When any codon is having information about particular amino acid, that is called as a sensible codon. But these three are having no information about any amino acid. That's why they are called as a stop or a nonsense codon. And if you go through some books, in some books, there another name is given. They have another name. This one, UAG, it has name amber. Amber, UGA, opal, and UAA, ochre. Amber, opal, and ochre. Right? So these are the other names for stop codon. Now, what is the reason why they are called with us such names? Out of these three codon, stop codon, in fact they are stop codon. Out of these three stop codon, which was the first to be discovered? It is UAG. UAG is the first stop codon to be discovered. And who discovered this? This one. Two scientists. One is Epstein. And another scientist, Charles Steinberg. So Epstein and Steinberg, these are the two scientists. They discovered first stop codon UAG. Okay. Now, why they called it as an amber? Because both of these scientists, they had a friend. His name is Harris Bernstein. This Epstein and Steinberg, both they had a friend. His name is Harris Bernstein, and this name is Bernstein. It is a German word. It is German word, and in a German language, Bernstein means a color. And what is the name of that color? Amber. So in a simple manner, let me tell you in a simple manner, these two scientists they discovered first stop codon, and they wanted to name this first codon with their friend name. Okay, their friend was Harris Bernstein, and this Bernstein term is German language, where Bernstein means amber. That's why they call it as amber. And if you search in some sources, amber, you will come to know it's a type of color. Okay, and since it is a type of color, they maintain the same pattern. They named the remaining two stop codons with a color pattern. So this is about the stop codons. Hope you know that what is the meaning of stop codons. Stop codons, they do not code for any amino acids. Okay, they stop the process of translation. That's why they are called as a stop codon. And students, this George Gamow, he said that the genetic code is a triplet. Yes, we should agree that. Because all the genetic code, they are triplet. But there was a problem. He said only theoretically. Theoretically, by means of assumption. Right? By means of a hypothesis or assumption, he said that genetic code is triplet. But science need practical proof. Okay? So when George Gamow said that a genetic code is a triplet, there was a need of a practical proof. That practical proof was given by 3 scientists. There are 3 scientists. One, Hargobind Hargobind Khorana is a one scientist, and another one is Nirenberg. His complete name is Marshall Nirenberg, and third one is Severo Ochoa. These are the 3 scientists. What is their contribution? They proved practically that the genetic code is triplet. How they proved? You see here, first scientist is Hargobind Khorana. We Indians should feel proud about this man because he is Indo-American scientist. Okay, this Indo-American scientist Hargobind Khorana, right? He developed a method. This fellow developed method of chemical synthesis of RNA with known sequence of nitrogen bases. This is the contribution of Hargobind Khorana. So what he did, this man Hargobind Khorana, he developed a method. What is the use of that method? It is a chemical method where you can synthesize RNA artificially. See, sir already explained a process called as a transcription. And what happens during a transcription? In a transcription, RNA will be synthesized in a cell. Within a cell, RNA will be synthesized, right? But this fellow developed a chemical method, a procedure where RNA can be synthesized, right, without using any biological system. And he synthesized RNA with 2 types. One is called homopolymer, and another one is called copolymers. Right? There are 2 types of RNA are being produced by Hargobind Khorana. One is homopolymer and another one is copolymer. What is the meaning of homopolymer? This RNA homopolymer, it has only one nitrogen base like U, like this. When RNA is made up of, there are 4 nitrogen bases, AUGC. In case of RNA, there are 4 nitrogen bases, AUGC. But he synthesized artificially, chemically RNA having only one nucleotide. That is what called as homopolymer. And what is the meaning of copolymer? Again, in another type of RNA, he used 2 nitrogen bases, Cu, like this. The RNA which is having only U means uracil, it is called as a homopolymer. Where RNA which is having Cu, right, cytosine and uracil, this is called as a copolymer. By producing such RNA, our Hargobind Khorana said that genetic code is a triplet. That is his contribution. Hargobind Khorana. So what you can expect about his contribution? He was the man who introduced chemical method of artificially synthesizing an RNA. Okay, there are 2 types of RNA, that is the homopolymer and copolymer. That is about Hargobind Khorana. Now you see second person, Nirenberg. So what is the contribution of this man Nirenberg? he developed a method where you can prepare protein without using cell. Okay, students, in a cell, protein synthesis will occur. Synthesis of protein in a cell, particularly in ribosome, that process is called as the translation. After this genetic code, after genetic code and mutation, we come to know what is a translation. So here at present, what we need, the meaning of translation is protein synthesis. Where it occurs? It occurs in a cell, particularly within a cell in a ribosome. But this man, Heisenberg, he developed a method where you can synthesize a protein without using a cell. Such method is called as cell-free system of protein synthesis. Right? So this man, Heisenberg, developed a method that is called cell-free system of protein synthesis. And what he did, he used the same homopolymer and a copolymer developed by Herbovic Karuna, right? He used this one, uracil. So he used an RNA, this is the RNA, having only uracil. Such RNA is called as a homopolymer. Then he allowed this RNA to synthesize protein, and he got to know that when RNA having a 12 uracil, see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. So there are 12 uracil. When the RNA is having a 12 uracil, these three will go for one amino acid, that is the phenylalanine. See, phenylalanine. Here also phenylalanine. So Heisenberg come to know that when there are 12 uracil, how many phenylalanine are produced? 4. Then he said that since the 12 uracil are there, they are responsible for 4 phenylalanine. So each of phenylalanine amino acid is coded by 3 nitrogen bases. That is what we call as triplet. Again, he used another method, another RNA, that is the copolymer. You already know what is the meaning of copolymer. It is made up of 2 nitrogen bases, that is the Cu, like this. Yes, so this is called as the copolymer. Why it is called as a copolymer? Because it is having combination of 2 nitrogen bases. Earlier one it had only 1 nitrogen base. Now it is having 2 nitrogen bases. That is the cytosine, uracil. So when it is used for the protein synthesis, Heisenberg got CUC. You see here, CUC code for what? CUC code for leucine. Next 3. UCU, serine. Again, CUC means leucine. Again, UCU means serine. Right? He come to know that when a copolymer is used where there are 12 nitrogen bases, out of 12, 6 are cytosine and 6 are uracil. How many amino acids are produced? There are 4 amino acids. Okay. By this method, Heisenberg come to know that amino acids are coded by genetic code, and each genetic code is having combination of 3 nitrogen bases. Now the last scientist, Severo Ochoa. So what he did, Severo Ochoa is a scientist from Spain. He is a Spanish biochemist, and he discovered an enzyme. Name of that enzyme is polynucleotide phosphorylase. polynucleotide phosphorylase. It is the name of the enzyme. Polynucleotide phosphorylase is the name of the enzyme discovered by Severo. What is the specialty of this enzyme? This enzyme produces RNA. It produces RNA with a definite and defined nucleotide sequence. Right? So what is the specialty of this polynucleotide phosphorylase enzyme? This enzyme can produce a RNA. That too RNA having a definite sequence of nucleotide. What is so special? It's not special, right? Because you already studied an enzyme called as the polymerase enzyme. DNA dependent RNA polymerase enzyme. Where you studied DNA dependent RNA polymerase enzyme is used for synthesis of RNA during transcription. Now that enzyme is synthesizing RNA. This is also synthesizing RNA. What is so special? There is one difference. What is that? DNA dependent RNA polymerase enzyme it need a template. Okay, I hope you know what is the meaning of a template. So it need the help of template. But here, speciality of this enzyme is it is synthesize RNA with a defined nucleotide sequence. Template independent manner. What does it mean? It means that this enzyme synthesizes RNA with a known sequence, nucleotide sequence, without using a template. Hence, it is called as a template independent manner. Right? And this enzyme was, what is the name of the enzyme? Polynucleotide phosphorylase is the name of the enzyme, and it was discovered by Severo. Hence, this enzyme is also called as Severo enzyme. You can call this as polynucleotide phosphorylase, or you can call it as Severo enzyme. And students, because of contribution of these three scientists, Khurana, Nirenberg, Severo, these are the three scientists. They practically proved that genetic code is triplet. This is the first property of genetic code. Still, there are many properties of genetic code. First property of genetic code is triplet. Now, let us see second property of genetic code. What is the second property of a genetic code? Genetic code is unambiguous. Right? It is a second property of genetic code. Okay? Genetic code is called as unambiguous. So what is the meaning of unambiguous? Unambiguous means you can call it as specific. What is the second property of a genetic code? Genetic code is called as specific. There will be no ambiguity. Ambiguity means this or that possibilities, but there is no possibilities. They are specific. Why genetic code are called as specific? You see this table. In this table, you see any one codon. We will take this codon only. U. This is U. Code for which amino acid? Phenylalanine. And just go through this table and find out whether in addition to phenylalanine, does this U code for any other amino acid? Right? Here, if you see this table, you will find U code for only phenylalanine, no other amino acid. It means particular codon particular codon will code for particular amino acid, that is what called as specific or unambiguous. Let me try with another example. You take this very famous codon for us, AUG. AUG code for methionine. You just go through this entire table and try to find out, does this AUG code for any other amino acid other than a methionine? Never. Okay, this is what called as genetic code as unambiguous, or you can call it as specific. Now, let us see third property of genetic code. Third property is genetic code is degenerate. Degenerate. Genetic code is degenerate. So what is the meaning of genetic code is a degenerate? Very simple. It's very simple. Let us see with the help of example. Just to see glycine. Whether glycine is coded by only one codon? No. See here. Glycine is coded by 1, 2, 3, 4. It means a single amino acid is coded by more than one genetic code. That is what called as degenerate. What is the meaning of a degenerate? Sometimes a single amino acid, one type of amino acid is coded by more than one codon, then it is called as degenerate. You can take still other examples. Say for example, leucine. Leucine is coded by, see how many codon? 1, 2, 3, 4, 5, 6. It means sometimes a single amino acid is coded by more than one codon, then it is called as degenerate. Both are exactly opposite. Unambiguous means particular codon will code for only particular amino acid, but a degenerate means sometimes a single amino acid is coded by more than one codon. Okay, and say for example, there are amino acids which are having only one codon. Okay, say there are two amino acids. There are two amino acids. Out of 20 amino acids, there are two amino acids which are having only one codon. What are those? One is methionine and another one is tryptophan. Say for example, methionine amino acid having only one codon. You just search the entire this table, you will find nowhere again this methionine. Methionine is having only codon. In addition to methionine, there is another, this one, TRP, means what is the full form of TRP, tryptophan. Tryptophan is also having only one codon. So methionine and tryptophan are having, these are the two amino acids having only one codon. Now there are some amino acids, they are having two codons. They are having two codons. What are the amino acids? Glutamine, glutamic acid, okay, asparagine, aspartic acid, here I am writing phenylalanine, tyrosine, histidine, cysteine, and finally lysine. So totally how many amino acids here I have written? 1, 2, 3, 4, 5, 6, 7, 8, 9. It means there are 9 amino acids. 9 amino acids. And each amino acid is having each amino acid is having how many codons? 2 codons. Let us check. For example, histidine you take. Histidine. How many codons are there for histidine? 2. You search entire table, you will find nowhere this histidine again. So it means histidine is having how many codons? 2 codons. Again, we will take this tyrosine. How many amino acids are there for tyrosine? Again, tyrosine will be having 2 codons. See? You can check again this asparagine. Asparagine. See? Here, asparagine is having 2 amino acids. It means these 9 amino acids, they are having more than 1 codon. How many codons they are having? They are each one of amino acids, they are having 2 amino acids. I mean 2 codons. And still there are some other amino acids. Still there are some other amino acids which are having 3 codons. 3 codons. Example, isoleucine. Here I am writing isoleucine. Okay. 1, 2, 3. How many codons are there for isoleucine? 3 codons. Right? Only 1 amino acid for this example. Example for 3 codons is only 1 amino acid, that is the isoleucine. And even there are some amino acids, they are having 4 codons. 4 codons. What are those? Here I am writing valine, threonine, alanine, proline, and glycine. So valine, threonine, alanine, proline, glycine, these are having how many codons? 4 codons. If you want to cross check, you take a valine C. 1, 2, 3, 4. It means totally there are 5 amino acids. 5 amino acids. These 5 amino acids are having how many? Each of the amino acids is having how many codons? 4. For example, just now we checked valine. Let us try one more. Proline. Just see here. 1, 2, 3, 4. Proline is also having 4 codons. And even there are some amino acids which are having 6 codons. 6 codons. So what are the examples for amino acids which are having 6 codons? 6 codons. Leucine, serine, arginine. Okay. So leucine, serine, and arginine. These are the 3 amino acids. Okay. Having how many amino acids? Having this 6 codons. 3 amino acids. So 6 codons having the amino acids are leucine, serine, and arginine. 3. Let us cross check. We will go for leucine. Let's find out leucine here. 1, 2, 3, 4, 5, 6. 6 codons. Right? If you want, you can go with this arginine. Here, arginine. 1, 2, 3, 4, 5, 6. So what is the meaning of this? It is the third property of genetic code. First property of genetic code is a triplet. Second property of genetic code is called as unambiguous or specific. Why it is called as specific? Because each genetic code will code for one particular amino acid. Okay. And third property of genetic code, which is the third property of genetic code? Genetic code is called as a degenerate. Why it is called as a degenerate? Why it is called as a degenerate? Each of the amino acid having a more than one codon. Okay. So when each of the amino acid having more than one codon, it is called as a degenerate. And student, I have one question for you. You just try to find out the answer for this. If you find the answer, you can post in a WhatsApp group. My question is, just now we discussed genetic code is degenerate, means each amino acid is having more than one codon. But there are two amino acids. There are two amino acids which do not follow third property of genetic code. Means there are two amino acids which do not follow that genetic code is degenerate. And your duty is to find out that two amino acids which do not follow degenerate property, and if possible try to post in a group. And this question was once asked in examination. Which of the amino acid, which of the following amino acid do not follow degenerate property? Okay, so that is all about today's class. In the next class again, we shall continue with this properties of genetic.